there was very important material from chapter 13 we didn't talk about at all. That had to do with linked genes. There is a big detailed study in your chapter looking at passing on what we call linked genes. That's genes that are close together. Can I get it a little quieter here? Can we make it a little bit quieter, please? Thank you. Linked genes are where we have genes that are close together on the same chromosome. In that case, when they're being passed on to offspring, they tend to be passed on together. They will sometimes get split up by crossing over and put on different chromosomes, but the closer together they are on the same chromosome, the less likely that is. So that's something where this idea of independent assortment of genes breaks down when you have genes close together on the same chromosome. So I am going to give you some panoptica material on that, chapter 13. You should look at, and whatever we don't do right now with chapter 14, I will also make a panoptica lecture. I recommend viewing both of those before we do the test. With that said, let's start with DNA here.
Doing what? Independent assortment.
There was a question for a long time about what genes were made out of. We didn't automatically figure that they were made out of DNA. We knew, getting back in the 1800s, that you had material that was being passed on in this mitosis process, and obviously that material had to do with affecting the abilities of cells to do different jobs, affecting their genetics. But the problem is that a chromosome is about half DNA and half protein. It's all those histone proteins and others that are helping to organize the scaffolding of the chromosome. If we go and isolate chromosomes and do a chemical analysis, your answers come back saying DNA and proteins. Both of those are being passed on. So which one was actually carrying the genetic information? Clearly it was the chromosomes, but which part? So we didn't get that settled until 1952, after a lot of arguing about this and a lot of studies that were done. And there were several experiments that led up to this being the answer. Then the year after that, we worked out the structure of DNA, and we were kind of off to the races with biotechnology. So early experiment with this, done by Griffin in the 1920s, he's showing that there was some kind of material that could actually be absorbed by living cells, taken from dead ones, and absorbed into the living cells, and giving them the ability to make some different proteins. Some kind of material could be absorbed by the cells. And what he did was experiments with bacteria. Bacteria are able to actually absorb material from their surroundings. And so we had two different versions of these Streptococcus bacteria. Ones that, under the microscope, they came to call rough ones, and others that looked smooth. The rough ones didn't do any harm if they were injected into a mouse. The mouse would be healthy. But if they injected smooth ones into the mouse, they would die. So there was some sort of genetic difference between these two versions of the strep bacteria. What Griffith did was kill the smooth bacteria with heat, so they were cooked. And then he had these cooked smooth bacteria mixed with a solution that had some of the harmless rough ones. And the rough ones had an opportunity to absorb materials from the surroundings, and they absorbed something which caused them to take on the deadly characteristics. So they were picking up something that came from these dead smooth bacteria and turned them into killers. It looked to Griffith as if what was being absorbed was a mix of DNA and proteins. That's the chromatin that chromosomes are made out of. So here's the experiment. He's got a mouse, injects it with living smooth cells. The result is a dead mouse. He injects a different mouse with living rough cells. The mouse is healthy. He kills the smooth cells by cooking them at high heat. Then he injects the dead smooth cells into the mouse, and the mouse is healthy. Those are the cells that would have killed the mouse, but now that they've been cooked, they don't do it anymore. But if we incubated living rough cells with the dead smooth cells, and then we inject that solution in here, those living rough cells have transformed, and now they're able to kill the mouse. They picked up something from the smooth cells. So there's a further experiment done by Avery. Avery is trying to tell exactly what it was that those smooth, or that those rough cells were absorbing. So he's continuing the whole thing, trying to do a more detailed analysis of exactly what it was that was causing the change. So he does some further processing. He digests RNA, DNA, and proteins separately. He uses different enzymes that can either break down proteins, or break down DNA, or break down RNA. And what he's then finding out is whether a rough cell is able to be transformed into a killer if it's absorbing DNA or RNA or proteins. And what he narrows it down to is that they only get transformed into the killer cells if he doesn't break down the DNA. He could break down proteins, he could break down RNA, but if he didn't break down the DNA, they would be transformed. So he's thinking that genes are made out of DNA. A lot of scientists think, okay, that proves it. Genes are made out of DNA. Not everybody's convinced. Basically, they argued about the methods. They weren't sure that he had this well enough controlled or, perfected as a study. So it looked promising, but a lot of scientists weren't totally convinced yet. Hershey and Chase are the ones that really definitively nailed this down. They're taking advantage of the fact that there are viruses that infect bacteria. We call those bacteriophages. That means actually eater of bacteria. They will eat a bacterium from the inside, forcing the bacterium to churn out copies of viruses until it explodes. So these viruses are putting their genes into bacteria, and they're made entirely out of proteins and DNA. That's what these viruses are made out of. They're extremely simple structures. And what Hershey and Chase were able to do was use radioactive isotopes of phosphorus and sulfur. And what they were doing was tracking where the radiation went. They made a population of viruses where they either were grown in a medium with radioactive sulfur, or different ones were grown in a medium with radioactive phosphorus. Now, where does sulfur show up in the biomolecules? We looked at a few chapters ago. What sort of organic molecules would have sulfur? Some amino acids. 2 of the amino acids have sulfur. Does DNA? DNA does not have sulfur. What about phosphorus? Does that show up in any of the amino acids?
Mm-hmm.
It does not, but it is in DNA. So basically, this is a label for proteins. That's a label for DNA. And what we do then is grow bacteriophages that either have labeled proteins or have labeled DNA. Let them infect the bacteria and see where the radiation goes. So these viruses are injecting their genes into the bacteria. We want to see if the bacteria get radioactive or not, and from which source. So here we have a bacterium. It's being attacked by these bacteriophages, about 20 or so in the picture. They are busy injecting their genes in here. Question is, will they be injecting in radioactive sulfur or radioactive phosphorus? That's a bacteriophage, as we now understand it, where we have all these parts shown in black that are protein, and the blue has the DNA. These things don't even look alive to me. They look like devices, like something we would build. DNA that's made out of the nucleotides, including phosphates, but again, doesn't have sulfur. And so what happened, I'm sorry, getting back to this, what happened was the sulfur did not go into the bacteria. Instead, it was the DNA with the phosphorus that did. So this experiment really did definitively prove that genes were made out of DNA, but it took until 1952 to get that worked out. Now, further work leading toward developing the model of DNA, the double helix, comes from work with these, the bases that are in the DNA. DNA has 4 of these kinds of nitrogen-rich bases. This is one of them, that's cytosine, but there's 4 total. 2 of them are small bases that we call pyrimidines, thymine and cytosine. They're both rings like this, both with several nitrogen atoms. And then there are 2 that are large. These are the purines, double rings, and these have a lot more nitrogen in them. So you'll have, in this case, the C's and the T's, the small ones, the A's and the G's, the large ones. And this guy, Chap, he studied many different species looking at their DNA. And so he had things like an octopus, a human, a chicken, various other things. And he was finding over and over that out of all the DNA that's in there, they have the same amounts of the purines and pyrimidines. So if you're looking at the 2-ring and 1-ring bases, they got equal quantities of those, and specifically equal numbers of the A and T bases and the G and C bases. So that's suggesting that a 1-ring base is pairing up with a 2-ring base, and specifically it's going like this: A pairing with T, C with G. Chap's rules. And so that is suggesting something about the structure of DNA, that there is some sort of pairing going on between these bases. So you end up getting the same amounts of those, same amounts of those. You might look at a bulk sample of DNA and find that 20% of the bases are A's. That means 20 there, and 30 there, and 30 there. If you know one of those, you're able to calculate the other 3, given Chap's rules. Well, Rosalind Franklin used a lot of this information, and she did work with high-energy X-rays to develop important information about the DNA molecule. This is once we were in 1952, 53, and we knew how important it was. This was where genes were located. She's doing this work to try to figure out the structure of DNA. It's work that should have won her a Nobel Prize, but she didn't live that long. By the time they gave out a Nobel Prize for this work, she had died from ovarian cancer. So she died at about age 37 or so. You know, just very cruelly cut short. And so the guys that got the Nobel Prize were these 2, plus her boss. So Watson and Crick are the famous ones that we hear about with the DNA structure. They're the ones that finally put it all together, putting together Chap's work, Franklin's work, and so on. They came up with the famous double helix, and they figured it out. And I mean, it was a brilliant thing to do, but it wasn't just them. So you end up with this double helix, where we have a spiraling structure that has 2 backbones running along, and that's where we have nucleotides joined together, sugars connecting to phosphates, connecting sugars to phosphates, and so on and so on. That gives a scaffolding that holds the bases in position in the middle of the molecule, running down the center of the molecule. And there we have A's and T's lining up and pairing and sticking to each other, and C's and G's sticking to each other. These dimensions are all things that Franklin worked out. All of these measurements, like the nanometers, the diameter of the molecule, and so on, that's all the stuff that she worked out from here. And getting in there in more detail, we find this arrangement. We have a sugar phosphate backbone running along like this, where you have a nucleotide right there. Here's another nucleotide, another one. The nucleotides all have a phosphate, a 5-carbon sugar and base. And one phosphate connects onto the sugar of the next nucleotide, and then that one connects to the next sugar, and so on. Notice when you're looking at this, is this strand oriented the same way as this one? Look at the way these sugars are set. Are these oriented the same way? This one is actually upside down compared to that one. It's what we call anti-parallel. These are running parallel but in opposite directions. And that's part of the genius of where Watson and Crick put this together. They realized you had these two strands, but one of them was upside down compared to the other. That makes it so the A's and T's pair up, and you have weak attractions between this base and that one. You form two hydrogen bonds, kind of weak, flimsy bonds between these two bases. And then between the C and the G, you form 3 hydrogen bonds. That's a little stronger connection. It happens because of the anti-parallel arrangement with this, and altogether it becomes the spiraling double helix that everyone hears about. Yes?
What is the 5 and the 3 and 3?
The 5 and 3 are just referring to which end is which, and I will actually show you that here in a bit. I think we'll get that far. But it's having to do with basically which carbon, the numbering of the carbons that are in these. That is known as the 5' carbon, and this one is known as the 3' one. So we'll see that, I think, here in a moment. So we get this base pairing. We always have a purine pairing up with a pyrimidine. That keeps this molecule at a constant diameter instead of bulging out or having to constrict. Anything where this doesn't happen is going to be a copying error of the DNA, which normally gets fixed by proofreading enzymes. Then we have the hydrogen bonding that is pairing up the A's with the T's and the C's with the G's. It becomes a weak connection between these. If you heat the DNA up to near the boiling point of water, about 95 degrees C, it breaks the hydrogen bonds, and these DNA halves will separate and drift away in solution. That's not very hot. Alternatively, you can have enzymes go in there and unzip this molecule, and that's the way we talk about it. It's just like the zipper action. Pull down a zipper, open up pants, you pull it up, put them back together. That's essentially what happens with the DNA molecule. The fact that these are weak connections makes it possible to do that. If they were covalent bonds, you wouldn't be able to break this apart into two separate halves, and then DNA couldn't do its job. For DNA to do its job, these need to be weak connections, and that's just what we have. Here's the numbering. If we're looking at one of these ribose, or deoxyribose sugars that we have here, it's a 5-carbon sugar. We number these 1', 2, 3, 4, and 5', and it has to do with which one of these is pointing toward the end of the molecule as far as the 3' and 5'. Makes a big difference that we're not going to be getting into here today. Notice on the other strand, it's pointed the other way. So right now, if this was the end of the molecule, that would be called the 3' end, and then this would be another 3' end if that was the end of the molecule. The primes come in because the bases get their own numbering, and so we'll have like a 1, 2, 3, 4 carbons there, and then they put the primes here to tell those apart. Let me show you one more thing here, and then I'm going to give you the thing to work on for the rest of the time. This is about the most important thing to leave off with. If you have an existing 1/2 of the DNA molecule, one strand where we have, say, nucleotides joined together, making a polymer of nucleotides. So imagine this is just half a DNA molecule, and you want to build in the other half. Well, that's what happens when we're in S phase, in interphase. Remember in S phase, we are actually making 2 copies of DNA from 1 copy. What happens is 1 copy unzips into 2 separate strands, and 1 strand serves as a template to build up the other strand. If you already have a T right there, you know that there has to be an A base lining up with that. A C has to go there, a G has to go there. And so you only need to have half of a DNA molecule to build the other half. That's critical when you're looking at the cell cycle and duplication of DNA. So imagine you already have that strand, and maybe you've already built this much of a new strand. You've already put in several new bases, and those are paired up with the bases that are there. Now we want to come in with a new nucleotide and add it in there. Usually that would take an ATP molecule to go and try to assemble things together. Like if you're building, say, amino acids into a protein, you have to use ATP each time to make the connections. You don't have to use a separate ATP molecule. What actually happens is you come in with a triphosphate version of the base. You've got this nucleotide coming in, it's the triphosphate version. And that already has the same energy as an ATP molecule. So the molecule that's coming in and being added onto this chain already provides the energy needed to do the assembly. So you end up getting the joining between this nucleotide and that one. The energy to do that comes from the phosphates that are already there. You end up just keeping one of those and rejecting then 2 of them. I don't think this, yeah, there it shows it. That shows this diphosphate coming off by itself, which then has to later be reconnected to another nucleotide. But the point with this is that we don't need to use separate ATPs to power this assembly. These nucleotides provide their own power as long as they're coming in the triphosphate form. The next one here, what nucleotide would have to go here? If that one's a G, it'd be C. It would be a C nucleotide that would have the triphosphate form, providing all the energy needed to put it into place. So that's a little variation on how we looked at synthesis of molecules powered by ATP. This is a bit different. Okay, let's see if we can do some things with what we've learned here. There's one.